today's video, we will address the possible role of biological, non-pharmacological interventions for ADHD.
So we'll speak about diet, probiotics, and the evidences that support this type of interventions.
As we have seen in a previous video, the pharmacological treatment for the core symptoms of ADHD, in particular psychostimulants, is highly efficacious, at least in the short term. However, the pharmacological treatment of ADHD does present with some limitations. Normalization is rare. Long-term effectiveness remains unclear. Adverse effects on sleep, appetite, and growth, although rarely serious and generally manageable, are common. And some parents and even clinicians have reservations about medications use. Therefore, there's been a lot of interest for possible non-pharmacological treatments for ADHD, including dietary treatments. Three main types of dietary treatments have been investigated in randomized controlled trials. First, restricted elimination diets. Second, artificial food color exclusions. And third, supplementation with free fatty acids. When assessing the possible benefits of non-pharmacological treatments, a crucial point is around blinding, which means that individuals involved in the study, including those who need to assess the severity of the symptoms before and after treatment, don't know if the patient is taking the active treatment or the control. For many non-pharmacological interventions, blinding is challenging, if not impossible. Some researchers have addressed this issue by looking at the so-called most proximal and most blinded assessors. Most proximal assessors are those individuals who are involved in the delivery of the treatment and hence may have expectations that influence their judgment when rating the severity of the symptoms. Typical example would be parents in parent training interventions. Most blinded are those that are more distal from the setting of the interventions and so less likely to be influenced by expectation bias. Applying this approach, a meta-analysis looking at non-pharmacological interventions for ADHD has found that when considering the evaluation of proximal assessors, restricted elimination diet had a significant effect on ADHD core symptoms with a high effect size, which however was driven mainly by two studies from the same author. Also, artificial food color exclusion and supplementation with free fatty acids had significant but small effects. However, when considering most blinded assessors, the effects of restricted elimination diet became non-significant, and the effect of supplementation with free fatty acid became even smaller, very close to zero. Artificial food color exclusion had a significant, moderate effect, which was, however, reduced when restricting the analysis to trials without concurrent pharmacological treatment. Therefore, overall, there is no solid evidence of strong effects of dietary interventions for core symptoms of ADHD. This is why clinical guidelines, such as the NICE guidelines on ADHD in the UK, do not advise elimination of artificial coloring and additives from the diet or dietary fatty acid supplementation as a standard treatment for children and young people with ADHD. However, these guidelines recommend clinicians to ask about foods or drinks that appear to influence hyperactive behavior as part of the clinical assessment of ADHD. And if there is a clear link, they suggest to advise parents or carers to keep a diary of food and drinks taken and also of ADHD behavior. If the diary supports a relationship between specific foods and drinks and behavior, a referral to a dietitian should be considered.
Samuele has just summarized what dietary supplements or certain types of diet can or cannot do for ADHD. But when we talk about food, we are also talking about the digestive tract, digestion, and a major player that are the bacteria that populate our intestine. We are talking about the microbiota. For the last 10 years, genetic techniques has allowed us to identify the bacteria that populate an individual's intestine. There has been a great interest in the study of the relationship between the microbiota in the digestive tract, the effect that it can have on the digestive system, and the impact of the microbiota on the development and functioning of the brain. We speak of the brain-gut axis. The bacteria contained in the digestive tract can influence the brain in different ways. First of all, through a direct nervous stimulation by the nerve that innervates the digestive tract, which is called the vagus nerve. But it is mainly through two other mechanisms that bacteria in the digestive tract can influence brain function. The first mechanism is the change in secretion by the digestive tract of a number of neuropeptides, neurohormones that will stimulate or influence brain function during the growth of a child and adolescent. The second mechanism is an inflammatory action of the bacteria directly on the digestive tract, which in turn, through the secretion of inflammatory factor into the blood by the digestive tract, will influence the permeability of the barrier between the blood and the brain and have a negative pro-inflammatory effect during brain development. All of this sounds complicated, but it's actually based on a few simple findings. The first is that recent studies on mice have shown that pro-inflammatory factor can influence neuron population and their development, differentiation, and through these neurodevelopmental changes have a possible impact on brain function. As we have seen, the dopaminergic system is the basis of the system that modulates the symptoms of ADHD, particularly in terms of functioning, cognition, emotion, and behavior. There is now evidence that the microbiota can influence dopamine level in the frontal cortex and striatum of mice and rats. The second finding is that studies of the microbiota in the gastrointestinal tract of a large number of people with very large samples show that certain less common population of bacteria were found more frequently in the gastrointestinal tract of people with ADHD. While these observation does not constitute proof that changing bacterial population can cause ADHD, it does indicate that changing microbiota could be a factor that could either alter dopamine secretion in individuals or have a pro-inflammatory impact that would modulate ADHD expression. So these findings have naturally led researchers to try to modify the microbiota, in particular by prescribing probiotic supplements, which are live bacteria or yeast typically found in yogurt or in certain type of food supplements. These probiotics help restore the natural balance of bacteria in our digestive tract, which may have been turned off balance by either a digestive tract disease, unbalanced diets, or antibiotic treatments. However, to date, despite an attempt to prescribe probiotics to large samples of children, totaling several thousands children, there have been no convincing results on neurodevelopment, cognitive function, or expression of adaptive skills in children who have benefited from these probiotic supplements. So, in conclusion, it can be said that there is a strong rationale for the bacteria in our digestive tract to influence brain function, and some studies suggesting population changes in bacteria in people with ADHD. However, to date, attempts to modify the population of digestive bacteria composing the microbiota in order to improve ADHD have not been conclusive. But obviously, all these studies are recent, and there is still a lot of work to be done, and we cannot, at this stage, draw a final conclusion on the ineffectiveness of probiotic supplement on ADHD symptoms.
Likewise, the possible role of dietary intervention is still very limited, and we do need more evidence on their possible effectiveness.